Welcome to This Week in Oncology. This week we're covering 10 notable papers spanning long-term outcomes and biomarker refinement in hormone-driven cancers, resistance and new drug platforms in oncogene-driven lung cancer, chemotherapy-free strategies in lymphoma, and a cluster of papers about drug safety and how we read so-called negative trials. Let's dive in. We'll start with hormone-driven disease, where two papers in Annals of Oncology push in the same direction. Longer follow-up and better biomarkers change who we treat. The ENZAMET trial, reported by Zhang and colleagues, now has 8 years of follow-up in metastatic hormone-sensitive prostate cancer, comparing enzalutamide with a first-generation non-steroidal anti-androgen on top of testosterone suppression. The survival advantage has held. Median overall survival was just under 8 years with enzalutamide versus under 6 years with the older anti-androgen. And at 8 years, half of the enzalutamide group were alive compared with about 40% of the control arm, roughly a quarter reduction in the risk of death. Clinical progression-free survival at 8 years was more than doubled, at about 43% versus 20%. What I find most useful clinically is the granularity on late toxicity and durability. Of the 622 deaths, most were from prostate cancer, and those were clearly fewer with enzalutamide, while deaths from other causes were essentially identical between arms. So there is no signal that we are trading cancer deaths for cardiac or neurologic ones. Grade 3 or higher cardiac and nervous system events were the same rate in both groups. Falls were more frequent with enzalutamide, but at a low absolute rate. Median treatment duration was nearly 5 years with enzalutamide versus under 2 years with the anti-androgen, and among the third of participants still on drug at cutoff, almost 90% were holding the full 160 mg dose. That is a strong argument against reflexive dose reduction for fatigue in patients who are tolerating therapy. The second paper in this theme may change a conversation you have every week in the breast clinic. Kalinsky and colleagues went back to RxPONDER, the trial in hormone receptor positive, HER2 negative, node positive breast cancer, with a recurrence score of 25 or less, where chemotherapy benefit was confined to premenopausal women. They measured pretreatment ovarian reserve hormones in over 1,500 participants under age 55. Estradiol, progesterone, luteinizing hormone, and follicle-stimulating hormone told them nothing about chemotherapy benefit. Anti-Mullerian hormone did. Among the 64% of women with anti-Mullerian hormone at or above 10 picograms per milliliter, that is, preserved ovarian reserve, adding chemotherapy to endocrine therapy cut invasive disease-free survival events by more than half. Among the 36% of women with low ovarian reserve, there was no benefit at all. The point estimate actually favored endocrine therapy alone. Distant relapse-free survival followed the same pattern, and an ultrasensitive inhibin B assay was also predictive. Critically, anti-Mullerian hormone outperformed menopausal status, age, and every other hormone as a marker of who benefits. This is not yet a licensed assay-driven decision, but it strengthens the hypothesis that the chemotherapy benefit in these women is largely an ovarian suppression effect. And it argues that a young woman with biochemically depleted ovarian reserve is unlikely to gain from cytotoxics. Turning to oncogene-driven lung cancer, where the theme is what happens after our best targeted agents stop working. Facchinetti and colleagues, also in Annals of Oncology, profiled 590 patients progressing on targeted therapy using paired tissue and liquid biopsies. Among 312 patients progressing on first-line osimertinib, RAS alterations accounted for resistance in about 11%, with a striking enrichment for KRAS G12D and almost no G12C. In ALK-positive disease, RAS alterations were roughly 3 times more common after lorlatinib than after second-generation inhibitors, and across MET, ROS1 and RET-driven cancers, they appeared in 7 to 16% of cases. In patient-derived models carrying acquired KRAS G12D and G12R mutations, combining osimertinib with either a selective G12D inhibitor or a pan-RAS inhibitor was markedly synergistic in vitro and in vivo. 2 practical messages. Tissue and liquid biopsy were complementary rather than interchangeable. So if you are only sending plasma at progression, you are missing mechanisms. and the RAS inhibitor pipeline now has a rational, testable indication beyond primary RAS mutant disease. Alongside that, Cancer Cell reports a phase 1 trial of SWOG S1010, an EGFR targeting antibody-drug conjugate in 236 patients with previously treated advanced non-small cell lung cancer. Response rates range from about a fifth in EGFR wild-type squamous disease to roughly 46% in EGFR mutant disease treated with tyrosine kinase inhibitors, but not yet chemotherapy. In the heavily pretreated EGFR mutant group, who had had both a tyrosine kinase inhibitor and platinum, median progression-free survival was 7.6 months and median overall survival 19.4 months. Toxicity was almost universal and myelosuppressive. Grade 3 or higher events in just over half of patients, driven by neutropenia, leukopenia, and thrombocytopenia. So this is a payload with a real hematologic cost, though notably not the interstitial lung disease signal we watch for with other lung antibody-drug conjugates. Our third theme is chemotherapy-free treatment in lymphoma. In the Journal of Clinical Oncology, Lynch and colleagues report a response-adapted, entirely chemotherapy-free regimen for untreated indolent B-cell lymphoma. 42 patients, mostly follicular lymphoma and mostly stage 3 or 4, received 8 cycles of the CD3 by CD20 bispecific mosunetuzumab. Those not in complete metabolic remission then went on to polatuzumab vedotin plus obinutuzumab. Every patient responded. Complete response was 71% after mosunetuzumab alone and 86% at end of treatment, with 2-year progression-free survival of 89% and no deaths. Cytokine release syndrome occurred in about 2/3 of patients but was uniformly grade 1, and no one needed tocilizumab. The 4 progressions are biologically interesting, 2 from CD20 loss and 2 histologic transformations. This is a single-arm phase 2 study in 42 patients, so it does not displace chemoimmunotherapy or the watch-and-wait conversation, but it is a credible template for randomized testing of a response-adapted, chemotherapy-free first-line. Finally, a cluster of papers about safety, trial interpretation, and guidelines. Clinical cancer research carries a commentary from Alumcall and Ellis on the withdrawal of the EZH2 inhibitor tazemetostat after the confirmatory Symphony 1 trial found hematologic second primary malignancies, mostly myelodysplastic syndrome and acute myeloid leukemia in about 6% of treated patients, and none in the control arm. Their argument is that this is not idiosyncratic but on target. EZH2 is context-dependent, and loss-of-function states are recurrent drivers of myeloid neoplasia, so chronic pharmacologic inhibition may phenocopy them. That matters directly for ongoing phase 3 programs in prostate cancer, where exposure is long and latency-dependent risk accumulates, a reason to be vigilant about cytopenias and clonal hematopoiesis in patients on any epigenetic agent. In the same journal, Shahnam and Gounder revisit Brightline 1 in dedifferentiated liposarcoma, arguing that a trial reported as negative in fact identified 2 active drugs. Berger-Madlin produced a median progression-free survival of 8.4 months, and responses in about a fifth of patients with translational evidence of p53 reactivation. The trial failed largely because doxorubicin substantially outperformed historical expectations, which resets the benchmark for future sarcoma trials. There is a shared lesson with the third paper here. A phase 2 trial in clinical cancer research from Kelly and colleagues added granulocyte macrophage colony stimulating factor to pembrolizumab in 42 patients with advanced biliary cancer. The confirmed response rate was 12%, including 2 complete responses, and about a quarter of patients were progression-free at 6 months, but the study did not meet its pre-specified efficacy threshold. It was well tolerated, and paired biopsies showed increased CD8 T cells and antigen processing, so the immunologic hypothesis remains live even though this combination should not move forward as designed. And rounding out the week, the Journal of Clinical Oncology published 2 ASCO Living Guideline updates, one on stage 4 non-small cell lung cancer with driver alterations, and one on immunotherapy and targeted therapy for advanced gastroesophageal cancer, both worth a look given how fast both fields are moving. If you only have time for one paper this week, make it the RxPONDER Ovarian Reserve Analysis in Annals of Oncology. A single pretreatment hormone measurement outperformed menopausal status and age at identifying which young women with node-positive, low-recurrence score breast cancer actually gain from chemotherapy. And that is a decision we make constantly. Here are the key takeaways from this week in oncology. Enzalutamide survival advantage in metastatic hormone-sensitive prostate cancer persists at 8 years, with fewer prostate cancer deaths, no excess non-cancer mortality, and most long-term patients holding the full dose. In premenopausal breast cancer with a recurrence score of 25 or less, low anti-Müllerian hormone identified women who derived no benefit from adjuvant chemotherapy, supporting the ovarian suppression hypothesis. RAS alterations drive resistance in roughly 1 in 10 oncogene-driven lung cancers. They are enriched after the most potent inhibitors, and tissue plus liquid biopsy together find more than either alone. A chemotherapy-free, response-adapted, bispecific-based regimen achieved high complete response rates in untreated follicular and marginal zone lymphoma with only grade 1 cytokine release syndrome. And on interpretation and safety, EZH2 inhibition carries a plausibly on-target myeloid malignancy risk that demands vigilance in long-duration trials, while both Brightline 1 and the biliary immunotherapy trial remind us that a missed endpoint is not the same as an inactive drug or an uninformative study. That's your roundup for This Week in Oncology. The full transcript and references are available on the episode page in your Audio Scholar library. This is an AI-curated summary. For clinical decisions, always consult primary sources and current guidelines. See you next week. If this weekly briefing is useful, follow the show in your podcast app so new episodes arrive automatically. And a quick rating on Apple Podcasts or Spotify helps other physicians discover the show. This is an automated summary generated by artificial intelligence, which can make mistakes. Always review the original source materials.